Hello everyone. In this video, we want to continue talking about the application of the biotechnology. We have studied what is artificial selection and what is genetic engineering. Now we want to talk about cloning. Cloning in biotechnology, or let's start to define what does a clone mean. A clone is an organism cell or piece of genetic material that is genetically identical to the one from which it was delivered, and you're talking more than one copy of it. Okay, cloning can be used to make copies of segments of DNA found at crime scene. Cloning is also used in genetic engineering. So cloning, you can find it for crime scene, for example, where they use it, and cloning is also used for genetic engineering. Actually, in 1966, scientists cloned the DNA from one sheep body cell to produce a clone, which they named Dolly. And they make a new sheep, which is what they named this sheep Dolly. Sorry. Here I'm going to show you a video how they make this clone to produce this sheep. I want you to listen carefully and to understand how they clone this sheep. Listen carefully to this.
To Moo Math and Science. In this video, I'd like to talk about animal cloning, how they clone Dolly the sheep. Basically, in order to clone another animal, you take a cell from one, then you take an egg without a nucleus, and then basically you get an offspring.
Okay, I want to stop here. Cloning in general, you take a cell, okay, then you and what, an egg without a nucleus. Here it should be what, an egg without a nucleus. You know, the egg cell, they have a nucleus, but this one you take it from without a nucleus, so it becomes what, an offspring. Look at this again. Sorry for this.
Then you take an egg without a nucleus, and then basically you get an offspring.
Let's see how
it's a little bit more involved. Dolly was the first animal that was cloned, and Dolly was cloned almost 20 years ago. Here's what happened. Step 1 was to remove a cell from the udder of sheep number 1. They then took this cell back to the laboratory and starved it in order to force it into an almost embryonic state. Next, they took an egg from another sheep, we'll call it sheep 2, and removed the nucleus. Step 3, they used electric shock and fused these two cells together. Step 4, they implanted this fused cell into a female sheep. They actually fused more than 200 cells, and one developed into a full adult. Dolly had the same cells as the original udder cells. What did we learn from cloning Dolly? The main thing was that it helped advance stem cell research, and it proved that the nucleus of an adult cell had all the DNA to give rise to another animal. Dolly lived for 6 more years and then lived from a lung disease. Anyway, that's Cloning 101. Thanks for watching, and Moo Math uploads a new math.
So here, this is in general how to use cloning. Actually, they use cloning can be used copy segments of DNA in genetic engineering. So they can use it, for example, in culturing body cells, for example. So here they can use it in medicine, which is very helpful. This one to use. So this is about cloning. Okay. Actually, you have to put in your mind if we are talking about biotechnology. Actually, here you have to put in your mind there are many ethical, legal, and social issues related to the ability to use and change living things. So, biotechnology can impact both our society and our environment, and we must decide how and when it is acceptable to use certain biotechnology. We can think about things that help for our society and environment, not to think about things that can harm our environment. Okay. And here I want to talk about how do you feel about the following application of technology. I want to ask you this one. Changing the DNA of mosquitoes so they can't spread malaria, for example, you can use it. Or cloning an endangered species of animal such as the gaur. Okay, modifying corn and soybean DNA to make them resistant to plant eating insects, for example. You can use it for these things. Okay, listen carefully to this.
A clone is an exact genetic copy of another organism. In 1996, scientists used biotechnology to clone a sheep named Dolly. Sheep are normally produced when a sperm cell fertilizes an egg cell. But Dolly didn't come from a fertilized egg. Click each image to learn how Dolly was cloned. Dolly's cloning was done by first using a micropipette to remove the nucleus from a donor body cell. In Dolly's case, the body cell donor came from a kind of sheep called a fin dorset. The donor nucleus was placed into an egg cell whose own nucleus had been removed. The egg cell was from a kind of sheep called a Scottish blackface. A pulse of electricity was used to fuse the nucleus and the egg cell. As the fused cell divided, it developed into an embryo. The embryo was implanted into a surrogate mother where it developed into a lamb and was eventually born. Dolly looked exactly like the sheep from which the body cell was taken. Their genetic codes were identical.
Okay, so this is for cloning. I want you to move to page 166, 167 in your book. About the effect of biotechnology on society and individual. You have here an example, a glowing mosquito. What is the glowing mosquito? This is the larva, as you see here in the figure. This is the larva of genetically engineered mosquito. Its DNA includes a gene from a glowing jellyfish. Actually, they have inserted a gene from a glowfish to its gene, okay? That causes the engineered mosquito to glow. So that's why you can see it can glow, okay? Scientists hope to use this same technology to modify the mosquito's genome in other ways. For example, it is thought that the DNA of the mosquito could be changed so that the mosquito could not spread malaria. Okay, so here what is the effect on individual and society? If the mosquito could be engineered so that it doesn't spread malaria, many lives could be saved, right? The effects on environment: mosquito are a food source of birds and fish. Okay, you have to think about other organisms also, not about you only. So, are there health risks to animals that eat genetically modified mosquito? Do you think? I want you to think about this one here. Okay, I want you to look at the other page here. The other page. Which is about cloning. The cloning gaur is an endangered species. In 2001, a gaur was successfully cloned. So the clone named Noah died of the bacterial infection two days after birth. What is the effect of individual on society? how will we decide when it is appropriate to clone other types of organisms? You have to think about this, okay? Let's talk about the other figure. Or also here you have the effect on the environment. A cloning could help increase a small population of endangered species like the gar and save them from extinct extinction. Sorry. Okay. So also here I want you to think about this one. Let's do it as a homework for you to think about it, about the top plants. Okay, much of the corn and soybean grown in the United States is genetically engineered. The plants have a bacterial gene that make them more resistant to plant-eating insects. How that will affect the society, do you think? Um, I think if corn is more resistant to pests, corn will be more readily available for people to eat. Effects on the environment. How might this affect the insects that feed on corn and in turn the organisms that feed on the insect? You have to see these effects, okay? This is all about the application of biotechnology. If you have any question about them, you can send me your questions. Thank you for listening.